Right, thank you very much, Ove. I guess we all have heard and know what's happening in terms of climate change and the impacts on the reef. What I'd like to focus then on today is the increasing urgency to understand coral health and to come up with any ways we can to mitigate coral health and improve it in the coming years. What I'm going to talk about today reflects the work of many outstanding postgraduate students and colleagues. So I'd like to acknowledge up front that there should be many co-authors on this talk, and also that I can only outline very briefly a few of the results of our studies in these past couple of years. Nope. Right. All right, I think we've all dived at some point on reefs that look like this. Sadly, this reef, like many that we've known and loved, has suffered significant coral mortality recently. In fact, in this case, from a devastating disease called white syndromes. It was responsible for loss and mortality of about 50% of the plate corals in a very short period of time, about 5 years since 2004. I think this highlights, and it certainly crystallized my resolve to try and understand what does govern coral health. So what I'm going to focus on today then is trying to look to summarize some of the symbioses that do underpin coral health so that we can gain an understanding of how we might enhance it. And then secondly, to try and summarize some of the work we've been doing on drivers of coral disease and also to evaluate if we can actually manage some of these. Now, you've already seen these two graphs. Jamie showed them this morning and nicely set up the points that I would like to make. First off, with the Diath paper, this is the results that show from Mantito survey data that mean coral loss on the Great Barrier Reef has been driven primarily from hot storms and bleaching. But Mantito data, it makes it very difficult to identify factors like coral disease, whereas with the Osborne paper, which looks at coral mortality based on scuba diving surveys, it's clear that there are more factors that should be considered, and in fact, disease is one that does factor into overall coral mortality. The point that I'd like to add is that, in fact, we know that there are very clear links between disease and storms and COTS, particularly the injuries caused by these sorts of disturbances, and also bleaching is known to reduce the disease resistance of corals, and often you do get disease outbreaks following bleaching. So in fact, one of the questions that I'd also like to explore is, does disease contribute more to the sorts of coral mortality that have been recorded over the past several decades than would be indicated by these figures? Now, to my first objective. It's become clear in the last 5 to 10 years that corals are, in fact, a very complex organism. They are made up of a multi-way symbiosis that involves a number of partners. Some of the main players are Symbiodinium, here, the unicellular dinoflagellate, and we know graphically what happens when the symbiosis breaks down, the corals bleach, and as well bacteria. And just to provide some perspective, our health is dependent on bacteria that live in our guts and our various orifices, but whereas we have something like 10 million bacteria per centimeter squared on our surfaces, corals, in fact, have 10 times that amount, something in the order of 100 million bacteria per square centimeter. So what are all of these bacteria doing, and how might they function in coral health? Well, some very fascinating insights are coming from work by J.B. Reyna, who has completed a PhD just recently, co-supervised by David Bourne and Shri Mati at Ames. And he's been looking at various roles of DMSP metabolizing bacteria in coral health. Some of the most recent research will appear in Nature in the next couple of weeks. Because of the Nature embargo, I can't outline the details. I can give you some broad sketch of the symbioses, and indeed I can show you some of his earlier research, and together it does make a very fascinating story. Basically, it's been known for some time now that coral reefs are the largest producers of DMSP on the planet. And it's the Symbiodinium in particular that are producing DMSP, which then coral-associated bacteria are able to metabolize. Now, when they do metabolize the DMSP, they produce a molecule DMS, which is climate active. What it can do is actually provide condensation nuclei for clouds, so you can, it can enhance local cloud formation, which then in turn is able to shade corals, and therefore could potentially reduce thermal stress. The hitch being only where coral cover is high. But more than this, DMS, when it breaks down, it produces sulfur-containing molecules, which can then be used to produce an antimicrobial, and that antimicrobial then in turn can reduce coral disease. Furthermore, DMS is a very powerful antioxidant, and it's possible that it can then be used in the coral thermal stress response. So I think even with these broad features of the story, and there'll be more to be released in the next few weeks, but it's quite clear that these microbial symbioses with bacteria play a very important part in enhancing coral health. And more than this, it highlights some of the further health costs when these symbioses break down, either through, and the costs that then are represented by declining coral cover and bleaching. Now to my second objective, trying to determine some of the drivers of coral disease. What I will highlight just here briefly is that we have been doing coral disease surveys now for 10 years. We've been doing those in 3 sectors of the Great Barrier Reef, the northern, central, and southern sectors, and we've been doing them in 3 different cross-shelf positions. Just for an example, here in the northern sector, looking at inner, so replicate reefs and sites in inner, mid, and outer shelf reefs. We've been doing belt transects and have now looked at well over 1 million corals over the course of these 10 years. Now, this data has only recently been finalized, but I can show you a preliminary pattern. All right, so here's, for an example, coral disease prevalence in the 10 years between 2004 and 2013 for reefs on the inner, mid, and the outer shelf. And I think one of the things that is clear that disease prevalence overall tends to be low. Nevertheless, what I'd like to highlight that even less than 6% disease prevalence indicates that at least 1 in 20 corals has one of these 6 very virulent diseases. And diseases like white syndromes and black band disease, they can result in the loss of a whole coral within months. Brown band disease can result in tissue loss in the order of 10 centimeters a day. So these do, even though it may look like a small prevalence, it's one snapshot in time, and that is the harbinger of significant mortality. The other pattern that's clear here is that there is a difference in disease prevalence between the different shelf positions, with disease prevalence being greatest, interestingly enough, on the outer shelf reefs. Here, the ones shown in blue, and least here on the inner shelf reefs. But it does highlight the point that for developing baselines of disease prevalence, and I'll emphasize these are current baselines, obviously we're working within this last decade, but we do need to develop baselines that are shelf specific. And that's the first step then in determining what's driving these disease, and then to go on from there, how we could manage it. Just to give you an example of how we are calculating some of these baselines, working on the definition that a disease outbreak is any case where disease occurs at a time or a place that's more than you would expect, or occurs at a greater rate than you might expect, then we can use this definition to try and work out what we would consider to be a baseline. In this case now, I've said that we need to consider baselines at a shelf-specific level. So here are 3 reefs in the northern sector on the mid-shelf, and this shows the pattern over this time period. Oops, what's happened there? And what we've done is calculated a mean disease prevalence over the 10 years and one standard deviation, and then looked at some of these outliers. And in this case, these outliers that for black band disease correlated with very high thermal anomalies. And if you included those in the baseline, we would suggest that that would inflate it. So we have removed those outliers, then recalculated the mean and the standard deviation, and then included two other thresholds here, or 1, 2, and 3 standard deviations above the mean, and we're suggesting that that might represent 3 different thresholds that could be used for a management response. In this case, there would be 3 different levels of response that would be activated by this pattern of coral disease. A level 1 response, a level 2, and a level 3 corresponding to these levels that are 1, 2, and 3 standard deviations above this mean. These sorts of thresholds could be set at whatever level you would like, depending on the sorts of sensitivity that you would require to trigger a response. Going back to this pattern, there are many temporal and spatial patterns in the data, but one other pattern I'll highlight is that in 2004, these reefs were rezoned from blue, which allows fishing, to green, the marine reserve status, and correspondingly, there was a decline in disease prevalence over the years following that rezoning, particularly here on the mid-shelf reefs shown in this red line. To try and get at what the driver might be, or what the links might be between that rezoning and this decline in coral disease, we need to break down this overall disease prevalence into the individual disease types. And this shows now for one of the mid-shelf reefs that was rezoned in 2004, and it shows fairly clearly that it is this decline in sclerotic band disease, which is a ciliate disease, that's driving this pattern. And in fact, the same pattern was found in the two other reefs that were rezoned in 2004. It was a decline in the ciliate disease, sclerotic band, that drove the overall pattern. And in contrast, for the reefs that were not rezoned, this disease remained low throughout the period of the study. So what mechanisms might underlie this decline in ZEB with the rezoning? And I'll refer to SEB as sclerotic band now. Oops. We have evidence that from another study, in this case a study from Koh Tao in Thailand, that injury may be involved in driving the increases and decreases in SEB. In this study, we looked at the level of disease between high-use sites here in the black squares and low-use sites in the white circles at 10 sites around Koh Tao. Now, what I want to say is that Koh Tao is probably the most popular spot to learn to dive in the world. There are some 50 dive operators that work in a very small area, about 2 square kilometers around the island, 2 square meters. But you can see here that the dive boats do raft up, and you do get quite a huge concentration of divers in very small areas. And what we found was, if you look at high-use sites, these sites here in histograms in black, there's a very clear increase in the prevalence of disease compared to low-use sites. In fact, it's 3-fold greater overall, and it's particularly the sclerotic band disease, again, that stands out. Now, if we happen to notice that at one of the high-use sites in particular, in fact, the site that had also the highest levels of this disease, there are also large numbers of corals that were entangled in fishing line. I don't know whether you can see here, but there are fishing line entangled all the way through this colony. So when we looked at the number, or the percentage of colonies that were healthy in ones entangled in fishing line versus those that were not, it's quite clear that entanglement has a huge impact on coral health. And in particular, its sclerotic band that is the main disease that we saw escalated. So here's the number of something like 80% of corals entangled in fishing line had sclerotic band disease, whereas something only in the order of 10% that were not entangled had the disease. And it was sclerotic band that was the major disease that increased with entanglement compared to all other diseases here. Interestingly enough, too, that when we looked at physical injury on these sites, there was an threefold increase in physical injury on transects with the discarded fishing line. So I think the linkages are starting to build that injury is an important driver of this particular disease. Now we have a second study which is also identifies an impact that causes disease that we can manage, this time from a study in Western Australia. And this was a study that looked at coral disease in association with the Gorgon project, which is the largest natural gas project in Australia's history. It involved the dredging and dumping of something like 7.6 million tons of sediment over 18 months to build a channel so that the ships could access the processing port on Barrow Island here in Western Australia. We used MODIS satellite imagery to visualize the dredge plume, and you can see here at one image what the dredge plume looked like. But based on looking at those images through time, then we looked at, or we categorized sites as being either high exposed to sediments, moderate or low exposure, depending on the number of days that they were exposed to the dredge plume, and then divided those into high impact, moderate, and low impact sites. Now, these disease surveys were led by Joe Pollock in collaboration with the Western Australia Department of Environment and Conservation, and what they found was that, again, there was an increase in coral disease from the low through to the high impact sites. Overall, there was a twofold increase in disease, but it was particularly white syndromes that increased. So this enormous increase in white syndromes on the high impact sites. In addition, when we looked at other indicators of compromised coral health, in this case also including sediment necrosis and bleaching, there was a six-fold increase in these sorts of indicators, and in this time it was sediment necrosis, this one here, this black part of this histogram that increased by 50-fold, bleaching also increased by 10-fold at the high impact sites. And now there are many different factors that could be driving this. We do know from previous research that thermal anomalies are linked with white syndromes. So we looked at a number of factors to work out what might be driving these differences in patterns in disease between the sites. And based on looking at 6 different metrics of thermal anomalies and also coral cover and dredging impact, and also predation by crown of thorns and drapella, a multivariate distance-based linear model showed that, in fact, it was the sediment plume exposure days that was the strongest disease driver. So I think this study, in summary, highlights that there are clear links between impacts like dredging and disease, and in particular white syndromes, and that there is some increasing urgency to manage these sorts of impacts in any sorts of coastal developments or dredging operations that might increase turbidity and sedimentation on reefs. All right, this brings me lastly to a good news story for coral health. We have evidence that the current marine park zoning does, in fact, enhance coral health. And this is a study that was led by Jolie Lamb, and we worked in collaboration with David Williamson and Gary Russ, who looked at the fish communities at the same sites. But when we looked at disease prevalence at, in this case, 21 sites in marine reserves, so in the marine national parks, which I'll refer to as reserves here, 11 blue zones, so in other words, habitat protection zones where fishing is allowed, and 10 conservation park zones where fishing is allowed, but there are gear restrictions, 1 hook, line, and rod. And compared that then, as I said, between the different zones. Here's a very striking pattern in terms of disease prevalence overall. So in terms of the prevalence of disease at fish sites, that was 4.1%, so 4 times higher than the level of disease that we saw at reserves. In particular, it was these first 3 diseases that were reduced in the marine reserves. The other 3 diseases, their prevalence did not really change between the different zones. These first 2 diseases are the ciliate diseases that we've been showing evidence that links that to injury. And I've just shown you that white syndromes can be linked to sediment. In this case, there were 31 variables that we looked at that might explain the differences in disease prevalence between fished and reserves, including fishing line, physical damage, differences in the fish communities, either the diversity or the abundance of fish communities. And in this case, the model, best fit model, showed that protection from fishing, in other words, the zoning, was the overall strongest predictor in terms of these patterns. There was also some correlation with coral damage, fishing line, and sediment necrosis. But overall, I think this highlights that if you reduce the impacts associated with fishing, that is good for coral health. This is in contrast to a previous study in the Philippines, which suggested that, in fact, it was intact functional fish communities that were responsible for improving coral health in marine reserves. So just to summarize and conclude, the work we've been doing over the last couple of years is increasing the evidence of the importance of microbial partnerships in coral health, and I think we'll hear a bit more about that from Bill and Madeline coming up. We now have a 10-year monitoring program looking at disease prevalence, which is providing baseline disease data for the Great Barrier Reef, and from that we can determine outbreak thresholds, and from that again identify disease drivers, and there are some clear drivers emerging that we can manage. And lastly, that marine park zoning can mitigate coral disease. And with that, then, I'd just like to acknowledge an outstanding and dedicated team of postgraduate students and surveyors, many excellent collaborators, and these are ones just for these particular studies that I've listed here, and funding bodies. Thanks very much.
So we have time for one question. Mike.
Yes.
So just a quick one. Looking at the percentage, like 5%, how rapidly does a coral die? Is it possibly die in like 1 month?
Well, yes, it can.
It could be an underestimate, sort of trying to say.
Well, yes, it can. So, I mean, I tried to the different diseases have different rates of mortality. White syndromes, it usually takes, it can be a couple of months. Brown band disease, it can be a couple of weeks. Black band disease, you can have, again, mortality, whole colony mortality over months. So the one snapshot in time, I think, underestimates what the levels of mortality. So 6%, I think, does represent significant mortality.
So looking at relative percentages like storms, disease, and all the rest, we have to calibrate at the same time.
Well, I think one of the points I'm trying to make is that we do have to factor disease into what we're looking at in terms of understanding the driver, or understanding the impacts of these various drivers on coral mortality. And indeed, I mean, we have really good data that suggests that crown of thorns starfish injuries result in disease, that storms, and I didn't have time to show you today, but we've seen increases in disease that continue for 2 years that correlate with the amount of breakage following storm damage. And finally, that thermal bleaching, certainly in the Caribbean, there's some beautiful examples of thermal bleaching resulting in disease outbreaks straight afterwards. Recover from bleaching, but die from disease. So I think disease does need to have a higher profile. Mm.